Welcome everybody to another one of our PHA4GE seminars. I'm Peter van Hersden from the South African National Bioinformatics Institute, and I'd like to welcome Colman from the European Bioinformatics Institute, who is a bioinformatician working on the ENA archival and analysis of SARS-CoV-2 data features. He completed his PhD at the University of Nottingham in 2020. Congratulations for completing a PhD in 2020 in Microbial Genomics. He's the chair of the Early Careers Committee at the UK Microbiology Society and also a member of that society's council. So yeah, the slides are loaded. Take it away.
Thank you. I'll just share my own screen because I've got my own slides. Where are you? Okay. Right. We are ready to rock. So yes, thank you for having me, and it's a great opportunity to be able to do this webinar today. So I really just want to take an opportunity to talk about SARS-CoV-2 at the ENA and everything we can do for the community and what we are doing at the ENA as well with respect to archival and analysis of SARS-CoV-2 data. So I thought I'd break this into kind of two broad sections, kind of an overview of just submitting to the ENA in general, if you're not entirely familiar with the process, so the processes that you can use to submit to us. And then also the second part is going to be talking about the European COVID-19 data platform, which is kind of the main feature of how we're presenting a lot of the SARS-CoV-2 data here at the European Nucleotide Archive, and also some of the new features that we're implementing through that portal. So to open it up, like I said, we're trying to start with a bit of how to submit and more what, why, and how of submitting to the ENA. So the ENA is one of 3 nodes that make up the INSDC, that is the International Nucleotide Sequence Database Consortium. The other 2 nodes being DDBJ in Japan and NCBI, which I think most people are generally most familiar with. And really all it means is that the data that is shared into those nodes, submitted into each of those nodes, gets shared and replicated throughout the nodes of the INSDC. But the ENA has a slightly different model, I guess, to the other 2 nodes, which is represented by this table. And this is the homepage of the insdc.org website, essentially. But I think it well illustrates the idea that when you switch to the ENA, it is a single service, it is a single team. It's not broken into sub-services. And as such, we have a different approach to how we've had to model how that data is archived in the ENA. And I'll talk about that now. And I think understanding our metadata model is a really important feature of actually understanding what it means to archive data with the ENA. I think it gives a very clear picture of the process that we go through when we archive data. And if users understand this better, I think it's a really useful tool for helping understand how to submit. So this metadata model is pretty simple, actually. It's nothing too complex. And each of these data types basically corresponds to an accession type. So studies are a very broad overarching accession. I think we're almost all familiar with study accessions as performers projects as well, we also call them. So these would begin with PHA4GEs as their accession number. And this is an overarching accession in which we house data, and it is responsible for understanding who's the owner of the data, what do you do with the data that's archived within a project, is it public, is it private, and it allows for us to understand and control what to do with the data that's held within projects. And often people will assign projects to papers. They'll say, like, this is the project for our paper on E. coli sequencing or something to that effect. And that's a sensible way of archiving data. And those are great ways of referencing data sets under project accessions, because all of the data will be under that one accession. The next major accession I think it's worth talking about are samples. So samples are a very important accession type within the ENA metadata model, because really it's telling us, what did you do? What did you sequence? What are you actually extracting data from? You know, and that's quite an important contextual metadata, because, you know, and we can't attach data unless we know what you sequenced. We can't assign runs to something unless we know where those runs came from in terms of what biomaterial did you sequence. Is it a sputum sample? Is it a colony that you picked from a plate? You know, is it a metagenomic sample? You know, did you sequence some sewage? It doesn't really matter, but we need to know what you sequenced in order to start understanding all of the other data around it. Runs is a very straightforward one, I think, because it's almost self-explanatory. Runs corresponds to raw read data, essentially. And this is one of the two fundamental data types that you can really archive, the other being analyses, which I'll talk about. And runs are literally just the data type itself, but they fall under this overarching associated metadata model, where they are associated with metadata contained within experiment accessions. And the experiment metadata is really for you to tell us, how did you generate those runs? You know, what sequencing platform did you use? What was your library selection? You know, what kit did you use? It doesn't really matter, but all of the metadata can be captured in experiment, and the runs are really just the data files themselves. And then collectively, they make up this raw reads archival point, where that metadata can get combined and is much easier for you to search and understand. The final data type that you can actually archive is analyses. And this really is what we'll talk about more really today, because analyses is genomes, annotated or otherwise, transcriptomes. There's quite a lot of different data types that you can archive under that one particular broad branch, but we won't talk too much about the other types. We'll focus mainly on genome submissions later on when we talk about archiving SARS-CoV-2 data, and runs being also the other main important data type that we archive here. So we have three submission methods or submission modes, I would say. So there's interactive, which is a kind of user-friendly graphical interface on the web that you can use. Web and CLI, which is a command line interface, as its name implies, and programmatic submission. And as you can see from this table, you actually can't submit all the different data types in any one method, but you can, if you become proficient at any two, would be able to submit almost every data type with good degree of proficiency. And I'll run through each of those different submission modes, kind of explain the logic behind them and what you'd be looking at, but very briefly, because I do want to get into the meat and potatoes of the talk, which is about SARS-CoV-2. So interactive mode, as you can see here, you can submit pretty much anything that's not an assembly or other analyses. You can submit template sequences, which are annotated sequences interactively. But depending on the size of your sequence, that mightn't be, or how many you're submitting, that mightn't be the best choice. This was updated this year. So there is an old Web and Interactive that some users may be familiar with. This has been superseded by a newer version, which is internally called Web and Submissions Portal, which gives you a much more friendly way of looking at things, and you actually get to see our metadata model represented in these interactive faces. And it's very straightforward of how to register samples, how to register a study, submitting reads. And we have also added these reports sections to this portal. So you can actually see what we see on the back end of the archival process. You can see at what stage your files are in the archival process, if they failed, what reasons they failed for, which I think is very useful for users to have a bit more verbosity in terms of their interaction with submitting data. So this is highly recommended if you're not entirely confident with submitting programmatically or in command line, but it isn't the most efficient way to submit if you have a lot of data to submit. But it is also the most user-friendly way to register reads, to to register samples and studies because it just makes a lot of sense when you're clicking in and reading things. The command line interface, again, is kind of like almost nearly like the inverse of the interactive. So you can't register studies or samples, but you can submit all your assembly types using the command line. And Web and CLI is actually just a Java program used to facilitate submission. So when you invoke it, it's a jar file. It's pretty easy to download, install, runs on up to, I think, Java 1, OpenGDK 1.8 anyway. And it's a relatively well-documented piece of software. And again, all the documentation for submitting to ENA is online at enadocs.readdocs.io. When you're using the Web and CLI submitting tool, there's really quite a few options, but there's only a few that you need to use to submit using the tool. Context, tell us what you're submitting to us. Is it a genome, transcriptome? Sequence here refers to annotated sequences specifically. Reads or taxonomic reference set, which I'm sure isn't probably of massive interest to a huge amount of users, but it's an important route of submission nonetheless. The manifest file is a rather simple text file which contains 2 columns, column 1 being a field and column 2 being field values. And I'll give you an overview of what I mean, an example of what I mean by that. And down here are some of your more important features. The submit option to actually submit the data. You can also run this in validation mode using the validate option. So you will actually get the pre-submission validation, and it will tell you if your submission could be successful if you chose to submit, which can be very useful because it means you don't actually end up submitting in parts. You know, if you submitted something as a bulk job, but only a couple of things failed, you'd actually have to go back and then resubmit 3 things or a couple of things separately, which can be a bit frustrating for users. So the validate is a very handy step without having to redo a whole job. You can also submit to our test servers, just as if you're experimenting with archival and experiment with tools using the test option. And you can use the ASCP option if you wish to use the Aspera CLI transfer protocol instead of FTP. Of course, that is every user's own choice. This is what I meant by a manifest file. Like I said, 2 column files, simple text. You need things like the study reference, the sample reference, assembly names, coverages. You can tell me what program you use to assemble your genome. This is an example for a genome manifest. Platform, Illumina, Oxford Nanopore. Minimum gap length. It can be set to at least 1, usually in most cases, depending on what you're submitting. Molecule type, and then the file type and the file name itself. So it's actually a relatively friendly way of submitting if you're comfortable enough using the command line, and can be easily scaled up programmatically if you have the skills. Programmatic submission is the final way of submitting. So you could submit all other types of analysis except assemblies. You can register studies, samples, and you can submit reads. Programmatic is the most advanced method of submission we have. It's the most bare bones method of submission. It requires users to create and submit XMLs directly. It is a very good idea to utilize the support of the ENA if you want to go down this route, and it can be very powerful when it's done right. But again, like I said, if you are unfamiliar with XML schema and how the data structures within the ENA exist, then I wouldn't entirely recommend this route unless you want support from the ENA side to help you develop your submission method. Now, on to submitting SARS-CoV-2 data. So we do have a dedicated Read the Docs page. If you do go to our Read the Docs, there is a dedicated SARS-CoV-2 section highlighting pretty much everything I'm going to talk about today. But I do encourage users to always go there. It hopefully contextualizes things a bit better because the rest of the docs are very general. But this is very focused, and it's the only case where we've ever written a focused submission guide for one organism. We also have a dedicated support team. So if you email virusdataflow@ebi.ac.uk, you will get through to either my colleague Zara or Ahmad, and they will be able to support your submission in whatever way they possibly can. So I highly recommend people to use that service where they are unsure of what to do about submitting. You do need to register a study. Again, and back to that metadata model, you need something to claim ownership over any data you submit to the ENA. So you need a study for it to be fallen under. So I always suggest to users, it's a good idea to go through the interactive mode, so the web and submission portal, because if you've never submitted to us before, you still need to register a Webin account to submit with us. And this is just a very simple way of registering your study information. I would say much the same for sample registration, but it depends on scale. And if you are a very high volume submitter, you've probably already been in touch with us about this. But sample registration, we do use a checklist system here at the ENA to try and maintain minimum metadata for samples. And the checklist we use for SARS-CoV-2 is called the ERC 000033 checklist. And I'll talk about what is covered by that checklist. We also offer a tool now, which is Python script. It is available to convert, if you've already submitted assemblies to GISAID and you have the metadata spreadsheet for that, this is a Python script which will enable you to just convert that into a spreadsheet for sample registration of matching ENA fields. So it takes a lot of the legwork out if you've already submitted to GISAID and want to submit to the ENA also. So I highly recommend people go to that. That is a public repository, by the way. Obviously, anything I do link here is public because that is part of the FAIR data process here. So I will try and share these slides as well. So all of this should be available to everybody. So this checklist that I just mentioned covers some mandatory fields here on the left. So these are things that you cannot submit to the ENA, like your sample will not go through unless you filled in these mandatory fields. And these are pretty straightforward: geography, host common name, IDs. These are like user IDs, health state, diseased, healthy, not applicable, not collected, host sex, host scientific name, collecting institute, isolate. Now, there are some fields that aren't actually mandatory, but we strongly recommend that you add to this sample when you're registering it, such as the organism scientific name, severe acute respiratory syndrome coronavirus 2, the taxon ID, as per the NCBI taxonomy, the collection date of when the sample actually collected, and this field sample capture status, which we've set it is controlled field anyway. You'll be able to drop down to that specific wording. So yes, that is the principle behind that sample checklist. If you do want to know more about sample checklists and how we use them to it's more to make the metadata very searchable and much more organized, which you'll see why that's really important in a little while. Like I said, submitting reads can be submitted by your preferred route. So if you want to submit those interactively or programmatically, interactively or through the Web and CLI program, that is up to you. It really depends on the volume. I would suggest the Web and CLI if you were submitting a high volume of reads at once, because interactive might handle it so well. Assembly submission is recommended by the Web and command line interface as well, or new SARS-CoV-2 REST API, which I will talk about shortly, because I'm sure many people will be quite interested in that. Don't forget to you can reference any previously archived raw data. So if you've already archived your runs and are only just getting around to submitting assemblies, you can actually reference those run accessions within your submission, which I think is really important for the integrity, because if someone wants to try and reassemble and re-examine different approaches to assembling a genome, it's important they know how what reads generated your assembly, so that they can try it for themselves. And it can be very important for various control data sets as well. So the Web API, this will be of very keen interest to probably high volume submitters of assemblies to the ENA if you are interested. So the documentation again is on the Read the Docs page specifically. This basically is two service endpoints of RESTful APIs, one validation and one for submitting. Again, this kind of works like the command line interface. The validate will tell you if your submission is going to go through all right without it actually being submitted. And the submit is the submit section. There is a test and production endpoint for both of those. So if you are just testing some software, testing submission, testing your methods, you can submit to our test server instead of the production server. The test server deletes any records after 24 hours, but works the exact same way as the production server. And basically, all you have to do is send those endpoints a JSON payload that looks like this. This is the schema for the JSON, and this is very human readable. And the asterisk prefixed fields here are the mandatory fields for submitting through this method. And here are the other fields that you can also use. So most of these are relatively self-explanatory. There are other fields within this, like TPA means third-party assemblies. So if you are submitting an assembly using publicly archived data that's not yours, you should set this to true because it just triggers an additional layer of validation. But this is very useful for programmatic submission of genomes. And I'll give you some examples of how you can use this programmatically. Here's a curl example, relatively straightforward using standard curl commands. And like I said, you just give it a single JSON payload. Now, obviously, the sequence field within this JSON is generally much longer if you're submitting a whole genome to us, particularly a SARS-CoV-2 one. But still, the benefit of having such a small genome means is that we are able to implement this type of this type of data accessioning. Because if we were trying to submit something any larger than 33 kilobases, this would be a very unwieldy field in a JSON file. And if you are of the more Pythonic frame of mind, as I am, you can do this using the request module within Python just as easily. And even better, I think, is you can use a list of JSONs if you wished to go down this route, which I think makes it a very powerful method of submission. So the Wellcome Sanger Institute piloted this when it was developed, so before we made this public, and they've probably been able to manage 30 to 40,000 submissions in a week using this method. So it is incredibly efficient if you are just looking to basically archive FASTAs, not annotated sequences necessarily, but FASTAs in particular is the benefit here. Bulk Web and CLI is a Python wrapper script that was developed by my colleague Nadeem Rahman. So we have, like I said, the Bulk Web and CLI Java tool. But this is a Python wrapper that allows you to basically do a lot of the legwork for you in automating the process. If you give it a spreadsheet, it will do most of a filled-in spreadsheet. It will do all of the submission for you through the single Python wrapper script. So it will upload your data to the appropriate place in Webin. It will use Web and CLI to do all the validation, prepare your manifest files. So if you are interested, that is again a public repository. It is Dockerized. It's containerized in both Docker and Singularity if you are into containerized software. And again, it's very, very efficient method of trying to ramp up your submission. And particularly, you can go down the route of submitting assemblies or reads using this tool in a very high throughput manner once you're familiar with it. And again, we're here for supporting anybody looking to use these tools. Please feel free to get in touch. A new feature that's worth mentioning is you can now claim your data in the ENA using ORCID. So if you have an ORCID, you can claim projects through this endpoint here. If you search for your project through that endpoint, you will be able to claim your data sets, which allows users to claim public data records in terms of just getting recognition for their contributions to open data. Now, finally, I do want to talk about briefly the COVID-19 data portal, really only for 3 or 4 minutes. This is a very long ongoing project, a very collaborative project across Europe. So VO is a partnership between these organizations, so MBLEBI, ELTE in Hungary, Erasmus MC in the Netherlands, and DTU in Denmark, which to basically create open pathogen sharing infrastructure. And the COVID portal has been a main feature of the work over the past year. There are other partners involved in maintaining this portal, like Elixir, in terms of providing data and infrastructure behind it. And it is hosted here at EBI and powered by a lot of EBI services. This is what it looks like when you hit the home page. So it is supposed to be a portal. You interact with a lot of different EBI services and a lot of different archival points through the portal. And as you can see, just from this homepage, there are far more records in the viral sequences section than there are anywhere else on the page. But it is a great way for interacting with the data that's archived within the ENA and the EBI in general. So you can explore sequences. So we have the data types here on the left, in this left facet. So you can explore all the sequences archives. You can view exactly what they're cross-referencing to within the ENA. Do they have samples, projects, proteins? Anything will appear here in this table. You get the metadata presented to you rather easily. Collection dates, country, center names. As you can see here, we've actually started running pangolin on all of the publicly archived sequences at the ENA. So we are also giving lineage assignments to the sequences that are submitted. And not only do they have the pangolin lineage definition, they also have the WHO definitions where available presenting also. We have a lot of filter faceting over here on the left. I'll go a bit more into what you can filter by in the sequences section. So center name is an important one. So as you can see, COG-UK, of course, submitting a huge volume of reads. Sequences, country, and importantly as well, coverage is a relatively new feature as well. So you can, if you're only interested in things that are between 0 and 20% in coverage for whatever reason, then we can easily present you a list of those files and accessions. You can also explore the raw read data that is archived in the same portal. And again, we have filter facets available to filter by instrument platform, library selection strategy, and even instrument model. So you'll notice that these are actually experiment accessions as denoted by the ERX or DRX prefix. So these aren't directly related to runs, like you won't click out and get to a run, but if you go to the experiment accession, you will see all of the associated runs with that experiment. Again, very useful. And I'll go back. You can actually download any table you want. So you can either download the full metadata or just a list of accessions, depending on how you filtered them, which can be useful for trying to download those data later. And I will talk about ways that we've developed to facilitate bulk download of data as well. There's a statistics page, which is pretty fundamental, pretty basic, really, but it's very useful to kind of have a representation of where you can see what is represented and by how much in the ENA in terms of data type. And finally, we have started doing systematic analysis on all publicly archived raw read data within the European Nucleotide Archive. These are accessible through this systematic analysis section here on the left-hand side. So when you're looking at data types, you will find the runs through which these analyses were generated. And we generate 3 different data types per run. So we generate a full unfiltered VCF. So this is just every single variant call without any care about what went into calling those variants. A filtered VCF, I'll talk about the process behind generating the filtered VCF and the consensus sequence shortly, and a consensus sequence for every run. These are all processed through one single workflow. Well, technically 2, but what I mean is that there is no discrimination between each sample or each run. They all go through the same process, irrespective of the context through which that genome was sequenced. These pipelines are publicly available and you can find them through the COVID-19 portal. But basically there's 2 pipelines responsible for generating these data. The COVID sequence analysis workflow developed by ELTE, our partners in Hungary, and the nanopore analysis workflow developed by Erasmus. So the COVID sequence analysis workflow is purely for Illumina data, and that outputs 3 different data types, like I just talked. about. The filtered VCF has a cutoff of, I guess it's referred to as allele frequencies in VCFs, but you know, not really allele frequencies. But yeah, the fraction of the reads that need to call the variant needs to be at least 25%. And consensus sequences we are also producing based off those filtered VCFs. The Nalapore analysis workflow at the moment only produces unfiltered VCFs due to problems trying to create universal cutoffs or filters in order to generate the other two data types. These are produced basically in fortnightly snapshots. So we've analyzed 600,000 reads at the moment, or samples rather. And these are all publicly accessible data. So anybody can pull these data, anybody can use them for their own purposes. They can always examine them, reanalyze. And these are the project accessions that they are archived under. Again, all of this is very easily accessible through the COVID-19 data portal. So I recommend if you wish to interrogate these data or utilize them, please do. That is what they are there for. I do want to mention that we do have a bulk downloader tool. So this is, again, another Java implementation. So you can download any of the data that's archived in the COVID data portal through this little slightly interesting command line Java tool. Kind of works very similar to most of our download methods, but you can specify your filters within the download tool. You can select what data type you want to download. Are you looking at the XMLs, the archived files? Do you want to filter? Do you want only by a particular country? This kind of thing. So if you are looking to really download a heavy amount of data, I would highly suggest going down this route. And it's highly scriptable as well. It'll generate scripts for you to download different types of data. If you wish to go down that route to keep your archived, if you want to keep copies of data up to date. One of the projects we are working on is trying to implement next-strain visualization, particularly based off the systematic analysis data. So we're hoping to have a public instance of next-strain visible on the portal in the coming months. And this also pertains to the final thing I want to talk about, which is private data hubs. So private data hubs are essentially pre-publication archival spaces within the ENA. So we will assign you a data hub and you can upload your data to this data hub privately. It will be accessioned, but we can offer various workflows to you if you wish to that raw data to be analyzed. This can be very helpful for people that don't have access to the infrastructure to analyze a lot of data. So you can upload your data, it will be processed by us. We will generate the outputs. So either using our analysis workflow for COVID data, we have a phylogenetic tree output. So you can get a NUIC file of your sequences. We are hoping to implement next-strain reports privately within data hubs as well. So you get your own private next-strain instance if you upload your raw data, and also hoping to offer lineage classification for data as well. Again, it is a pre-publication private space. So until you want that data to go public, it will stay private. It is purely as a way of encouraging users to start using public repositories and perhaps offer added value to archival of their data where we can. So do reach out if you are interested in the private data hub system. Now, I think I've probably talked for plenty long enough. Any questions, I'm happy to answer, and you can email me at my email down here, okaho@ebi.ac.uk. As I mentioned as well, we have the virus data flow email as well if you wish to have any questions, concerns about submitting to the ENA. Thank you.
Thank you so much, Colman. While I wait for people can put questions in the Q&A section, but while I wait for those to show up, I've got a question of my own. So you're saying you're running Pangolin, but I mean, of late, especially with some of the sublineages of Delta. Pangolin has been kind of flick-flacking around a little bit with the different data releases as they refine their models. So when you say you run Pangolin, are you going to rerun it periodically?
Yeah, we are rerunning it daily.
Okay, so then you always use whatever the latest Pangolin database of the time is. Yeah.
Yeah, exactly. So Pangolin should get updated in the morning if there's a new release of their version, and then we run it, I think, a couple of hours later on all the data. So there might be a slight lag behind in terms of the presentation, because that's, you know, a day or two before you catch up on whatever the previous release was. But yeah, it's a daily update.
And then I see there's a question coming in here. Yeah, so Soyan is asking, how do you handle security associated with data upload from an end user over the open internet? Yeah, what is your security arrangements like for that?
That's a very good question. I mean, look, everything is, you need the Webin, well, to upload data to us, you need a Webin account. So, you know, we have user authorization through that to upload to any of our services, or to our FTP endpoints or anything like that. So that's our main way of doing that. In terms of downloading data, there is no block to that. You know, there is no authentication required to access any of the data that's public within the ENA. But I don't know if that answers your question specifically, but.
Thanks. I'm sure there can be a follow-up if that is needed. But yeah, Curtis Capsack is asking, where can he find more information on the bulk downloading tool, the Java command line? Is there a GitHub repo for the tool? Is it in Bioconda?
There should be a GitHub repo. If you drop me an email, I'll double check. I'll send you directly. If not, it should be on the COVID-19 portal. It should be relatively recent in the news tab, that we've developed that, so it should link out to exactly where we have the documentation for that. Yeah, but do email me if you need sources for things that I didn't include in the slides.
And so yeah, if you could find the link for, because I had a look around the data portal and I couldn't find the links for the workflows that were in use. So I might just be bad at clicking things.
Yeah, give me.
But if you could share, that would be useful.
Yeah, they should be. Yeah, go on.
From my side, I've got a question. So one of the recommended pieces of information that you had in your upload, I forget the name of the specification, and was something with a lot of numbers in it, was an ID for what looked rather like a patient ID. Where's that going to go? Because, you know, I mean, especially in areas where there's a small number of cases, that does suggest that you could, I mean, it does suggest some kind of privacy concerns.
Yeah, so this is a free text field. We kind of encourage users to make sure that it's more of an internal pseudonymized ID rather than anything directly relating to patients. Where those IDs appear to be identifiable or recognizable data structures as patient IDs, it'll generally get picked up by someone on our side as erroneous, and we will probably suppress and reach out to the users. Because we are very conscious that we really do not want to be posting any identifiable information at the ENA. If you do have anything remotely human related, submit to the EGA, not us.
Yeah, so let me take another question from Wakasidin Khan. If we submit our data to GenBank, it will be synced with ENA, even if the data is SC2 data, right? So in other words, it doesn't matter which of the INSDC databases you submit to.
No, that's true. That's absolutely true. If you submit to GenBank, it will appear in the ENA and the DDBJ. One caveat I would add to that is there are cases. So we accept more data from a genome perspective than NCBI might do for SARS-CoV-2. So we don't validate data for how much sequence is in there, whether or not you have odd-looking deletions or frame shifts or anything like that. So there may be cases where if you submit to us, it'll be accepted by us, no bother, but it mightn't always be mirrored by NCBI because they don't like the look of the data. That's a relatively rare case. Does that happen? So yeah, in most cases, it would always be reflected in all the INSDC nodes.
And how long, I mean, how often is that sync happening? Daily, weekly?
Ideally, it happens almost daily up to weekly. There are occasional delays in how that sync happens and how the mirroring happens, but we have people to keep an eye on that and making sure that we don't really lag behind too much. Then in terms of the ownership, so who owns the data that's submitted to ENA? I mean, does ENA claim any ownership? What happens if somebody derives a product from data submitted to ENA? So the entire principle is that if you're submitting to the INSDC, you're accepting that data be public. You have ownership over that data in a sense that it is still yours. You are the person responsible for generating that data. And that's kind of the logic behind the project accessioning as well, that lets us identify people. And now you can have a much more personal claim to projects by linking your ORCID ID. But by having data in the public sphere, you are implicitly accepting that people will reuse that data. And that's part of fair data usage principles as well, is that it should be reusable if it's truly open. And I mean, if you do not wish to go down that route of having your data fully open, then don't know really. You can submit privately if you wish.
Yeah, I know there have been ongoing discussions around different data sharing models and things like that I think outside of the scope of this webinar, though. Yes, Curtis seems to have found what he thinks is the correct link. But Curtis, if you think you found the link, please post it. Yeah, then they are the workflows. Thank you very much. Those are what I thought they were when I was looking at them. So one of the things I was also wondering about that, do you record which version of the workflow is used in the analysis? Because now you're going to get this VCF and this consensus genome, but maybe then, next month you change the workflow.
Yes. You will see if you go into the metadata for the analysis objects we're archiving, there will be a field where we record the version of that tool. So then you can go back and double check that nothing's changed or something integral hasn't changed drastically.
Yeah, thank you. That's very useful. Then in terms of the hubs, so the hubs, if I understand correctly, the hubs are kind of like, if you've got a particular group of people, maybe one project, and they want to have an environment for working on their data, maybe they don't have their own computing environment, or maybe they're in different countries and they would like something, together. Is there any model for kind of sharing between hubs, or is it basically that like, you're going to work in this hub, and then once you're done working, you're going to publish it on the ENA, but you're not going to like really share between hubs?
Yeah, so basically the way the model will work. So when we register a data hub, we require a user agreement. So there will be an agreement has to be signed. There will be two classes of users. There will be data providers and data consumers. And data providers will be the only ones authorized to upload data to that project, but anybody who is a consumer will be allowed to use that project. There is nothing precluding you from having several projects across different data hubs if you wish to have an agreement in place where you have a data hub sharing data with one particular group of people and a different data hub sharing the same data for slightly different reasons with another group of people. However, we would obviously expect you to be transparent with everybody that you are sharing that data with, but that's not something we can necessarily control. We have not really a way of, the principle is that each hub is slightly isolated from each other. So there isn't a direct way of sharing data across the hubs, but we can link any private data to any data hub in which you are a data provider.
Okay, so because I mean the data hub is kind of a view on, because the actual backing store is your main databases, right?
Yes.
So of some kind. So the hubs are kind of like views with particular associated permissions on something. So that's why you can do it that way.
Yeah, exactly.
Just email the right people.
Yeah, which would probably be me.
See any further open questions, although I do have another one from my side. Have you had any people like trying to link, to do linked studies between the viral and the host bioinformatics? So to maybe say that, okay, these people sitting in the host side had some outcomes and things like that, and then maybe we can find some features in the viruses that explain this. Is there anything like that ongoing?
Yes, we do actually. Well, there is ongoing work with the EGA, and there is a European grant supporting the work behind this, because it is an important way to do it. But I can, let me have a look for exactly. I do have somewhere to point you to, because it's not within my remit. But yes, that is ongoing because it's an important part of the data.
Thanks. And while you're looking at that, can I just drop another question? So I see you computing the pangolin lineages. Do you also compute the Nextclade clades, or is that not on your agenda?
When we have Nextclade implementation sorted, then we will be. There will be a question about if we present that also within that same table. But yeah, it's ongoing work at the moment.
Sorry, I forgot to unmute. Yeah, so we'll just wait and see then.
Yeah, pretty much.
Apparently, there was a question from Anthony Underwood, but I'm just trying to see if I missed it in one of the chats. Jamie, if you have that question, could you ask it? Oh, hand is raised. I don't see raised hands for some reason. Well, how do you deal with that? Sorry, Jamie, can you deal with the raised hand side of things? Because I don't see any controls on my side to ask a question. Oh, there is. Okay, it was apparently a mistake after all. Sorry, I know the Zoom technology is like overwhelming me.
Yeah, so I have actually, so I've also put in the docs, but the cohort browser is the way we've actually implemented how you can explore linked data sets for human and viral data.
I see. So then basically, of course, the focus, because I mean the host data is going to be and the EGA, and you know those who have applied for and been given access to that data can then see the cross links to your ENA viral data. That kind of makes sense because that's the one that's more sensitive. Soin is asking about the functional annotation process. Is it manual or automatic? Sorry, Soin, Kim, could you perhaps just elaborate the question a little bit, because I'm not sure which function is you're talking about annotating. Yes, yeah, the raw sequence to VCF. So that is now starting to happen automatically, right?
Yes.
So in theory, if you are a sequencing provider, I mean, I don't know, could you tell me how viable it would be to upload the FASTQ, and instead of having to kind of compute VCF and consensus locally, to basically have the ENA just compute it and then people can use it. I mean, you said you're going to be integrating with NextStrain in the future and so on and so forth.
Yeah, so basically the way it's currently working, so the workflow is entirely automatic and works on public read data, that is SARS-CoV-2, and works from the most recent data backwards. So we're trying to catch up on what is a 1.4 million read backlog, but basically we will submit anything that is recently submitted. So if you submit something relatively recently, it will probably get picked up within the next 2 weeks, and the VCF and the consensus will be automatically computed and archived. Yeah, so like I said, and like I said, those workflows should explain most of what we do in terms of automatic, like low freq being our variant caller as is the favorite for everybody, and annotation of the VCFs then is done by SNPF, but not the SARS-CoV-2 version necessarily.
This is just the original SNPF.
Yeah.
Yeah, and people can see the pipelines at those links that you posted. They, for people who haven't looked, these are Nextflow pipelines. And then just Lynn.Trung is asking if you could just explain again how you handle frequent Pangolin version updates.
Yeah, like I said, we are updating, running Pangolin on a daily basis. So as sequences come in, then they get Pangolin lineage, but we'll also update Pangolin whenever it becomes available, if they make changes to their software, which will then obviously be run instead, and the updates should be seen daily.
And you would run that on all of the samples, so eventually you'd be running it on 1 million samples.
Yeah, so all of the sequences should have Pangolin lineages, unless they fail Pangolin for whatever reason, like too low coverage or whatever. But we are also hoping to run it on the systematic data as well. So when we produce consensus sequences, we'll start running Pangolin on those.
Oh, I see. So you start off by, when people have uploaded their own consensus sequences, but you're moving over to the systematic as well.
Yeah.
Okay.
Yeah.
Thanks for the clarification.
Yeah.
Sorry, just in terms of curation. So if a user finds a problem with a consensus sequence, then they just re-upload and get a new version, right? Of the consensus sequence.
Yeah, that was our that is our process usually. Yeah, we have a versioning system for consensus sequences. Yeah.
And in terms of the data, the data hubs, I mean, can you give me a sense of, well, what is the largest collection of sequences that people are processing through a data hub at the moment? I mean, are these being really used at big scale?
That's a very good question, actually. We have several pilot hubs. So Erasmus probably piloted the earliest data hub, and I think they have upwards of 30,000-40,000 sequences probably are through that. So there isn't much limit. We definitely recently migrated some of our computer architecture so that the data hub system should be relatively efficient. And like I said, there's plenty of support usually on the ground at ENA to help you with figuring out how long that take. We also have a Greek data hub as well with the Elixir Greek node, and they probably have about 10,000 data reads archived in that hub at the moment.
Okay, thanks. So it's definitely scaling up.
Yeah.
Okay, we're nearly at the top of the hour. So I think that unless people have pressing questions, we can bring this webinar to end. Thank you once again, Colman. I am also the co-chair of our infrastructure working group meeting. So sorry to people for not being more explicit about the fact that we were canceling today's Western meeting to have this webinar, but we will see you at the next infrastructure working group meeting in 2 weeks, those who are from the IWG. Yeah, thank you very much, Jamie, for organizing all the logistics and recording and all of those things. and Rangarai for doing the comms as usual. And we will see you at the next webinar or at your regular PHA4GE events.